This is Chapter 3, Part 2, looking at staining. I mentioned earlier when most commonly we use a bright field light microscope when we're viewing bacteria or other microorganisms, with the exception of viruses, of course, that are too small to be even seen with a light microscope. They are naturally not colored, and so usually we have to stain them to be able to see them. So staining is just the process of coloring microorganisms with a dye so that you can see the organism, or sometimes to see a certain structure of the organism. So the process to create a stained slide of a microorganism is that you first have to put the microbe on the slide. That's called making a smear, and it's just as the name implies. Then you have to fix the microbes onto the slide. You can either do this with heat or you could do this with chemicals like methanol. And that keeps the microbes attached to the slide, and it also kills them in the process. Then you proceed with staining as the third step. But if you were to stain prior to fixing, then your microbes would be lost. They would generally end up washing off the slide during the staining and washing procedures. That's why fixing is important. The other importance of fixing is that it makes it safer to handle those slides when the microorganisms have been killed, rather than if they're still alive on that slide and you have to handle that slide.
Microscopes are very useful tools to study specimens smaller than about 1 millimeter. Light microscopes and electron microscopes use lenses to focus light or electrons passed through a specimen to produce a magnified image. Both light and electron microscopists utilize stain to increase the contrast and visibility of the specimen under study, as well as to aid in the classification of these specimens.
All right, that was a very short animation. So the concept of staining is really based on chemistry. It's based on charges. So bacteria have more negative charges in their cytoplasm and on their cell wall. And so it's just the basis of opposites attract. So positive dyes are attracted to the negative charges of a bacterial cell, and therefore the positively charged dye will stain the bacterial cell. So this is what's most commonly used. It's a basic dye where the chromophore, the dye basically, the stain is positively charged. It's a cation. But we do sometimes use acidic dyes, which is an anionic dye or a negatively charged dye. And that, instead of staining the bacterial cell directly, will actually stain the background, because we know that like charges repel each other. And if the stain is negatively charged and the bacteria is negatively charged, the stain cannot stick to the bacteria. It'll be repelled by the bacteria, but it will stick to the background of the slide. And it kind of creates what's comparable to a dark field image, where you get a dark background, you get a stained background in this case, and the microbe appears unstained and lighter against the dark background. So when we do the process of staining the background rather than the cell, it's called negative staining. When we do the process of staining the cell with the positively charged dye, it's called positive staining, or it's also called direct staining, since you're directly staining the cell. So a simple stain is a stain that only uses a single basic dye, and so both positive and negative staining can be considered to be simple stains, because both only use one dye. However, as you see, it does say single basic dye. So most commonly when we say simple stain, we're usually referring to that positive or direct staining. But it does vary. Some textbooks, some lab manuals will group negative staining as simple staining as well. Others do not. So the purpose of a simple stain, whether it be the positive direct staining or negative staining, is to show you the bacterial shape and size and arrangement of bacteria. That's the information that you can get out of a simple stain. In some cases, when we do staining, we will use a mordant, which just think of mortar if you've ever done like tile work or brick work. It's like cement. It's like it makes it stick, basically. So mordants can help the stain to stick or help to enlarge a specimen by coating it and making it look larger.
Light microscopists utilize stains to increase the contrast and visibility of the specimen, as well as to learn more about the features of the specimen. There are three main categories of stains used by microbiologists: simple stains, differential stains, and special stains. Simple stains are made of a single basic dye that interacts with the biological sample and stains it. After treatment with the dye, the sample is rinsed with water, leaving the stained sample. Simple dyes are used to determine size, shape, and arrangement of cells. Crystal violet is a purple stain that stains most bacteria uniformly. Safranin is a red dye that stains most bacteria pink or light red. Methylene blue is another basic dye that stains most bacteria a light blue color. Some of these dyes are also used in more complex staining techniques, such as differential stains. Most of the stains used in microbiology are differential stains. Differential stains use more than one type of dye and can be used to distinguish between different structures. The Gram stain is the most frequently used differential stain. It's used to distinguish between Gram-positive cells, which have a thick outer cell wall composed of peptidoglycan, and Gram-negative cells, which have a thin outer cell wall. The procedure of Gram staining involves 7 steps, during which the samples are stained with crystal violet dye, treated with a mordant, rinsed, treated with a decolorizing agent, and counterstained with safranin. Gram-positive cells are stained a deep purple color from the crystal violet dye, but Gram-negative cells are decolorized and then stained pink by the safranin. The acid-fast stain is a useful differential stain because it only interacts strongly with a subset of bacteria in the genus Mycobacterium that have a waxy material in their cell walls, as well as the pathogenic strains of the genus Nocardia. The acid-fast staining method involves several steps, during which the samples are stained with carbonyl fusion while being heated over steam, cooled, decolorized with hydrochloric acid, and counterstained with a simple dye such as methylene blue. Some bacteria produce endospores. Endospores form inside the cytoplasm of the bacteria and are highly resistant to desiccation, heat, and radiation. Their cell walls are impenetrable to most chemicals. The endospore stain uses heat to drive the dye malachite green into the endospores. After heating with malachite green, the slide is rinsed with water to remove the dye. The slide is then counterstained with safranin. In the final sample, vegetative cells are pink and endospores appear green. The endospore stain is a useful tool in classifying bacteria. Special stains are stains that interact with special bacterial features or structures such as the flagella or capsules formed by certain bacteria. Negative stains stain the background, leaving cells colorless. These acidic dyes are repelled by the negative charges on the surfaces of cells. Examples of negative stains include eosin and nigrosin. Some bacteria use tiny structures called flagella for locomotion. Flagella are too small to be seen with a microscope, but they can be stained in a multi-step process involving the dyes pyrrose-aniline and carbofusion, and the mordants tannic acid and potassium alum. The number and arrangement of flagella are important in the classification of bacteria.
Okay, so let's look a little bit closer at differential staining. So we will have labs on these topics as well, but the lecture will proceed by, I'm not sure exactly, at least a couple weeks, I think, before we get to this topic in lab. So first and most importantly is the Gram stain. The Gram stain is the most important stain in microbiology because it divides bacteria into the two main groups of bacteria, gram-positive or gram-negative. And this is important because there's differences in antibiotic resistance and sensitivity of gram-positive and gram-negative bacteria, because what distinguishes these bacteria is their cell walls. And it's very common that the bacterial cell wall is the target of an antibiotic. So gram-positive bacteria have a thick peptidoglycan cell wall. Gram-negatives have a thin peptidoglycan cell wall, but they have an extra layer of lipopolysaccharides. After carrying out the Gram stain procedure, gram-positive bacteria will appear purple and gram-negative bacteria will appear pink, like you can see in the picture here. So like we just saw in the animation, this is the procedure for the Gram stain. So the first step is to apply the primary stain, which is crystal violet. All the bacteria would be stained purple at that step. Then you apply the mordant. The idea of the mordant here is to help the crystal violet stick. And that's because the mordant, which is iodine, it combines with crystal violet. It forms large crystals, large complexes, which get trapped in the peptidoglycan layers, especially the peptidoglycan layers of gram-positive bacteria, because they have more layers of peptidoglycan. Gram-negative only have a thin layer. But regardless, crystal violet, iodine form large crystals in both gram-positive and gram-negative cell walls. But the critical step is the decolorizer, which is an alcohol. So once you apply alcohol to the bacterial cell wall, this is where differences happen depending if they're gram-positive or gram-negative. Gram-positive bacterial cell walls, the peptidoglycan gets dehydrated and it actually further traps in the crystal violet iodine complexes. However, gram-negative bacteria, the alcohol actually dissolves the outer membrane of lipids, and then because the peptidoglycan layer is so thin, it does not trap in the crystal violet iodine. That washes out, and now gram-negatives become colorless at that point, like you can see here. Gram-positives kept the purple color, but gram-negatives become colorless. So then you have the last step, which is a counter stain. The counter stain is done to then put color onto these colorless bacteria, because we said it's hard to see colorless bacteria on a bright field microscope. We want them to have color, contrast to be able to see them. So we use safranin as a counter stain, Safranin's a red colored dye, as you can see here. However, you can see in the image, generally, we don't actually get red cells, we get more of a pink, because once you do water washes, because what they're not showing here is there is water washes between each step. Once you do the water washes, it's not a red color that is retained, but more of a pink color. So that's how you get gram-positive being purple and gram-negative being pink. The acid-fast stain is also an important stain, but not nearly as important as the gram stain. The acid-fast stain is really important to distinguish the mycobacteria and the nocardia. Mycobacteria especially, because that's where you have the tuberculosis species, which causes TB or tuberculosis, and leprae, which causes leprosy. So these bacteria, they have a waxy lipid in their cell wall that does not let the primary stain decolorize when treated with an acid alcohol. And that's why they're called acid fast, because they retain that primary stain. So you can see the steps here. Primary stain's carbolfusin. It's a red fuchsia color, really, but the book likes to just say red, but it's much more of a fuchsia pink color. All cells would stain that color initially, but again, the critical step is the decolorizing step. Now, non-acid fast bacteria will decolorize and become colorless. Acid fast bacteria will not decolorize. They will keep that primary carbolfusin color. And then again, because we don't want to try to view colorless bacteria under the microscope, we counterstain, in this case with methylene blue, to get non-acid fast bacteria appearing blue and acid fast bacteria appearing red. When this procedure was initially developed, it was actually developed not to distinguish two bacteria from each other, but rather to be able to distinguish tuberculosis from lung tissue. So lung tissue would stain blue, and the bacteria would stain that fuchsia pink color. So special stains, you have capsule stain, endospore stain, flagella stain. So capsules, they are produced by some bacteria, and they are an extra coating outside of the bacterial cell wall. But they're made of polysaccharides usually, and they're non-ionic, meaning that they do not take up stain. And so how would you then stain a capsule? You can't. It doesn't take up stain. So what you do is you combine a positive or direct staining method with a negative staining method to make the capsule pop out as an unstained halo around the bacteria. So what they've done here is they've taken crystal violet, that's the purple here in the center. That's a positively charged dye, so it's going to stick to the negative charges of the bacterial cell. And so that's the purple that you're seeing is the bacterial cell. And then they're going to take an acidic dye, a negatively charged dye. There's a variety of them. In our capsule stain procedure in our lab, we use one called Congo Red. In Manival's, it's a special capsule staining procedure. Those chemicals don't really matter at this point, but they're going to use a negatively charged dye that's going to stain the background. And it's that combination of the positive and the negative that then allows you to visualize the capsule. So the capsule itself is not stained. You can't see the capsule unless you do this special procedure where you stain the bacteria inside with a positively charged dye. You stain the background of the slide with a negatively charged dye, and you get this effect of this unstained capsule popping out. Endospores are a very resistant structure that only a few groups of bacteria produce. The main groups of bacteria is Bacillus, which your main pathogen of Bacillus is called Bacillus anthracis, which causes anthrax, and then Clostridium, which you have several species of Clostridium, but you've probably heard of tetanus and botulism and maybe gangrene and maybe C. diff. So these are all species of Clostridium, and they produce endospores. And these endospores do play a major role in the transmission of those infections, because these endospores are very, very resistant, and they can persist in the environment, and then they can get into somebody's body and lead to an infection. So we want to be able to see these endospores, but they're very resistant and they don't naturally take up stain. If you do a gram stain on this bacteria, what would happen with these endospores is they would be unstained. And you might be able to kind of see the outline of them. You can actually see it here in some of these where they didn't quite take up the stain. You see this little white hollowed out area in some of these rod-shaped bacteria. So that's what happens in a gram stain. But again, we don't like to have to try to see unstained structures under the microscope. So you have this special endospore staining procedure to put stain onto those endospores. It uses heat with the primary stain. The primary stain is malachite green. Use heat. That way the heat drives the stain into the endospores. Once the stain gets in the endospore, it doesn't wash out. And so we don't even need a special decolorizer here. The decolorizer is just water. You wash the slide with water, but the green will stay inside of the endospores. And then you counterstain with safranin, because the regular bacterial cells, which we call vegetative bacterial cells, all those pink rods here, they would be colorless. And again, we don't want things to be colorless under the microscope, so we counterstain with safranin to put color on the regular bacterial cells. And so what you end up with is the regular or vegetative bacterial cells are the pink-red color, and the spores are the green color. The last stain is a flagella stain. So flagella are very, very thin, kind of hair-like structures that bacteria can have that they use for movement or locomotion. And so here you have to use a mordant that will coat the flagella and make it thicker so that it can become visible. So this is a question about fixing and why fixing is important. Here's a question about microscopes. Which type of microscope can be used to view viruses? And here's a definition question. And lastly, a true-false about capsule and endospores.